Sir, I made you as the host. So
yes, sir. Yes, sir. Thank you.
Yeah, you will have control now. Yeah,
okay.
Thank you.
Okay. Thank you, sir. Thank you so much. Shall I start, sir?
Yeah, let's give 1 more minute and then we'll start.
Sir. Okay.
I think we can start. Yeah. Sir, I think you can start now. Yeah.
Okay, sir. Thank you. Thank you so much for it. So good evening, all of you, my dear students.
Good evening, sir.
Good evening. Good evening. So, last class, what we have seen, will you please summarize the points? What are the things? Vegetative reproductions. What are the vegetative reproduction is there? What is by vegetative? Propagules. Particularly the plant, it contains, what are they? Roots, stem, and also flowers and rhizome. That things already we discussed, isn't it? And also, two type of, what is that? Vegetative propagation is also there. One is called as natural, another one is called as artificial. Today we are going to discuss the remaining part is called as artificial method of vegetative reproduction. It is otherwise called man-made vegetative propagations. Okay, from this heading, there is a three things we are going to discuss one by one. Okay, first one is called as cutting, and second one is layering, third one is grafting. Okay, see one by one. What is by cutting? What is by layering? What is by grafting? See one by one. Okay, see. First one is called as cuttings. Cutting is the most common method of vegetative propagations. Cutting is also one of the common method of vegetative propagation. For the commercial purpose they were used. Okay, how it will be? How it should be? Is there, first one is the cutting can be made from the stem, leaves or roots, part of the leaves and thick can. What is meant by can? It's nothing but the sugar can. They denoted as a sugar can. Okay, the cutting made from the stem, whether it may be a stem, it may be leaves or roots or part of the leaves and the thick cans also they used for cutting purpose. Okay, and the third point is for these plant pots that are insect and disease free or selected. That is very, very, very important. Suppose we are not going to plant a disease one, whether it is an infected one, otherwise called as a contaminated one, because it's a commercial purpose means you have to select the superior quality and also good quality, then only it will be better for your, what is that, yield. Okay, then according to this part, cutting also made by three methods. What are the three methods? It depends upon their plant pots. Okay, one is called as stem cutting, leaf cutting, and also root cutting, and also it is produce a new plant by the method is called as vegetative propagations. As a result, you will get the new plant and also used for the commercial purpose. So, okay, once again repeat for you. Cutting is the most common method of vegetative propagations. Mostly it's a man-made, particularly the stem, leaves, roots, parts of the leaves and thick can also they were used. And for this plant pots that are insect and disease free or selected, this is very, very important. So many times they were asked in the NEET questions. According to the pots, cutting also made by three methods: stem cutting, leaf cutting, and also root cutting. Okay. Go to the next slide. See it. Stem cutting. You have to select the stem. The stem should be in how it will be. See, one by one. The stem cutting usually included a portion of the stem with some leaves, because it may contain one node or two node or three nodes like that. It contains some leaves and is usually 3 to 6 inches long. Okay, the stem should be how much, it is included a portion of the stem with some leaves also present in it. At the same time, the length of the stem should be 3 to 6 inches long. And then each stem should have minimum 2 nodes. That's why I told you. Each node, a little bit bearing the leaf. Okay. So stems also having, which one, leaves. Leaves also having nodes. Particularly tell us that each node consists of leaves, like that you told. Okay. So this is also very, very important point. Then only the vegetative propagation is possible. Without nodes, without what is that, leaves, it will not propagate properly. That is very, very important things. And then third one is the tip of the stem usually selected and often gives the fastest result. Do you know why it is gives a fastest result? Why they are selecting the tip of the stem? Anybody else? Anybody else? Why they are selecting the tip of the stem? Any students?
So,
no. Why? Yes.
so the tip have more meristematic tissues, so it divides more easily and therefore it grows faster.
Okay, very good. So because the presence of what is that, easily a divisible portion, because the presence of what is that, parenchymatous cells. Parenchymatous cells undergoes easily divide. Okay, not only that, and also maximum the auxin is one of the phytohormone. Hello. Phytohormone auxin is mostly present in the tip of the stem as well as the root. That's why they were selected. Which one, the upper part that is called a stem, particularly stem tip. The tip of the stem usually selected and often gives the fastest result. Okay. Example has given what are the things, we cannot, but at the same time, one more is that the seed is present, but it will take more time to get the yield. Okay, that's why they have selected which one, stem cutting. Take an example, drumstick. When you are planting the seeds, what will happen, it will give rise to entire plant. It grows as an entire plant, and finally you will get the, what is that, yield. It will take how many years? 4 or 5 years or 10 years, sometimes it may take. Okay, suppose you are you're choosing this type of methodology, otherwise called as vegetative propagation wise. Likewise, stem cutting is a better method. Okay, you will get the yield within short period, within 6 months. Okay, you will get it. Okay, so what are the examples they have given here? Coffee and grape, citrus, sugar cane, bougainvillea, and also China rose. These are the examples of vegetative propagation, particularly done by which one, stem cutting. Stem cutting. Okay. Okay, good. Next. So this is a stem cutting already given here as a sugar cane. See that it is up to the 3 to 6 inches, and also having the nodes. Nodes also have which one, it has a, what is that, leaves also there. Okay. When it placed in the favorable condition, what will happen, automatically they provide, what is that, the buds, and buds also grow into a vegetative of leaves. It is called as new leaves, and also new roots also originated. Okay, so next diagram you can see it very clearly. Okay, this is called a sugar cane. It's very, very important for the commercial purpose. Is it? No, this stem should be how is it is? It should be a disease free. That is very, very important. So many times they were asked in the NEET questions. They were asked. Okay. Next, see that stem cutting of any one of the plant. The upper part, otherwise called as tip of the shoot system, is make a cut and placed in the another part. You will get the entire plant like this. Okay, so these are the another methodology. Take an examples, table rose, and also so many plants is there. Okay, drumstick is also there, rose, jasmine, grape, whatever it may be. These are the plants used as which one stem cutting, used for the vegetative purpose and also commercial purpose too. Okay, go to the next one. See, leaf cutting. So leaf is also already but it's also having the which one vegetative propagation properties. Okay, here leaf cutting. See the first one. Leaf cutting consists of the leaf or the leaf blade and a plant are formed at the base of the leaf or petiole. So leaf cutting is also made whether it may be a leaf blade. Blade means outermost surface is called as blade. Okay, and sometimes the leaf base is also having a small bud-like structures near the petiole. What is by petiole? Leaf attached with a small stack is called as petiole. Already you know, isn't it? Okay. The leaves support development of tiny shoots and buds. Leaves also produce the what is that? Take an examples of which one the bryobillum. Already I told you the bryobillum is also one of your leaf. What is that? Cutting methodology. Okay, you will get the new plant. How it will be grow? The leaves also there is a small tiny buds present in the surface of the leaves. It is called as a small shoot buds. The small shoot buds also what is that? What is that? It will grow as a entire plant when it undergoes the maturation process. After the maturation, it detaches from the parent. And it fell onto the where is it in the favorable condition. As a result, the formation of new plant. It grows a new plant. Okay. See, these buds may begins to form a roots at their base. As these shoots break off from the original leaf, they fall. That is, original leaf is nothing but the mother leaf. Okay, they fall on into the ground and take root. Fall on into the ground. So the ground means what is the meaning of the here? What is the meaning of this ground? Means favorable condition. Then it will grow. Okay, otherwise it will not grow because when the favorable condition is there, the plant as well as other things. Take an example: leaf, stem, root, or flowers. All these components, all these vegetable parts will grow into a entire plant. It is also used for the vegetative propagation. Okay, and you take a root. Example: Streptococcus and Pecnonia. These are the two things has given, and one more is there very, very, very important. Bryobillum is also known as mother of thousands. Why it is called as mother of thousands? For example, take an example. Any one of the bryobillum leaves, it produces more than 100. What is that? Leaf buds. Otherwise called as shoot buds. That's why each one it's considered as a mother. Okay, that's why it's mother of thousands. They have even a name is called as mother of thousands. Okay, so see the next slides. So this is the leaf. This is called as a mother leaves. The mother leaves, when it placed under the favorable condition, what happened? Correspondingly root formation also takes place. This is called as a root, and this is a newly formed leaves. Likewise, they have showed 4 or 5 things, 5 samples. Okay, go to the next one. So this is also another one. These are all single leaves. Single leaves cut into 3. What is that? 3 samples, otherwise called as vegetative parts. Okay, each one it's placed under the favorable condition. Each one it produces a root. Okay, produce a root. Correspondingly, the plant also will grow. Okay, next one. Third one is called as a root cutting. Already roots also having a which one the propagation properties. It has the ability to grow as a entire plant. Some herbaceous plant such as blackberry, raspberry, citrus, tamarind, acanthus, and papavera flax can be propagated from the root cutting. take a small piece of root from that particular plant already mentioned there, just take a samples and place in a separate pot or separate place, whether it may be garden or somewhere else in your house. Okay, the root also, what is that? It produce the leaves, and also primary and secondary roots also produce and correspondingly the leaves also produce, and it is also used for the commercial purpose. Okay, see the next one. See, these are the root. So the particular portion is removed from the mother plant, the root is cut, and after that it may be placed into a favorable condition. These are all are considered known as tissue culture, because it's a simple leaves also. What is that? When you are placed into the nutritive medium, it grow into a entire plant. After the growth of the root as well as the 3 to 4 leaves. Okay, this condition is called as, what is that? Vegetative propagation. After that, it may be transferred from the where to where, the lab to the normal land place. That condition is called as hardening. What is the meaning? When you are going to study the tissue culture, that time you may know these type of things. Okay, these are points. Okay, this is called as root cutting. And then go to the next one. So layering. So layering is also another methodology. Okay, cutting, and second one is called as layering, and third one is called as rebot. Okay, see, and also grafting. Okay, layering. Layering means, what is the meaning of layering? The development of root on stem while the stem is still attached to the parent. Okay, take an example. See, any one of the plant, if present as it is. Okay, just remove the bark of the, what is that, plant. Any stem, you should select it and then take a small blade and cut into 2 pieces. Okay, and completely remove the bark only. Okay, and remove the bark and add some amount of, what is that, whether it may be a bunch of what is that, soil, and cover it. Okay, cover it and with the polythene paper and tie it properly and put it into the 2 or 3 holes. After a few weeks, what will happen? You will get it. You will see the root originated from the removed bark removed area. So it is present as it is. What is that meaning? Parent is also present. Parent stem is also present. While the stem is still attached to the parent, the plant is called as layering. Okay, after the root formation, we kept the plant from the parent and it is maybe placed in the another pot. Okay, so this is called as layering. So the layer is a rooted stem following detachment from the parent plant. Detachment from the parent plant. A soft basal branches is defoliated in the middle where a small injury or cut is made. I will show you the next slide. You can understand better. Okay. Later, as the root develop, the branch or layer is separated and planted. Example: rose, jasmine, and grapevine, and also lemon. So these are all are considered getting also layering. Already you know a little bit. Cutting also same, because grapevine and lemons, citrus already told it. Here the lemon has mentioned, isn't it? Okay, lemon also we may, what is that, layering, we can do the layering process also. Okay, go to the next one. See this one is called a simple layering. Simple layering means, take an example, second one. This is the parent plant. This is it. This is the parent plant. The parent plant, the branches is the branches also, what is that? You have to make a small cut here. Okay, small cut. You place into the, what is that, soil. Okay, and cover with the soil. What will happen? After few weeks, you have to pour some water. What will happen? Automatically in this area there are so many advantageous roots also originated. After the root formation, what will happen? You have to make a cut here. Okay, after the root formation, you have to make a cut here and remove the plant and place it into the favorable condition in another pot like this. This is called a simple layering. This is called a simple layering. Parent is also there attached stem, you have to make a small cut here and buried under the soil, you can get the root after a few weeks. Then cut the plant branches and placed into the favorable condition. You will get the new plant like as a parent. It is also type of layering. Okay. Go to the next one. This is called as compound layering. Compound means more than one. Okay, here already we saw the first first slide. Only one layering is possible. Here there are two layers there. Sometimes some plant they have a, we can do the more than one layering is also possible is that. Okay, so this is the mother plant. Mother plant is also attached with the soil. The other branches, it may be just like a bend like structures, and you have to place in the under the water. I'm sorry, under the soils, buried under the soils. The each and every node, branches nodes also develop the roots. Okay, you have to make a cut here and also here also. This one and this one also. You will get the entire plant. This one is grow as a knee plant, and this is also a knee one. You will get the set of plantlets. Okay. So this is called as compound layering. And see that these are the three things. Okay, this is the three things. The same single branch is there. Single branch also they bend into 3 times. So you know about the here the plantlets also originated. So as a result, the root also formation is here. You have to make a cut here, and also you made a cut here, and also here also. So you will get the 3 plant. And separately. So this is called as compound layering. So these are also one of vegetative propagation. Suppose if you want to more plantlets, you can do this type of methods. Okay, I will get it. So many plantlets. Okay, see. So I told this is called as a mount layering. Mount layering just touch the soil only. So, okay, here what will happen is take a parent plant, the branch touch the soil. So you have to place any one of the weight on it. What will happen? Automatically you have to add some amount of soil and also water also. As a result, what happen? Here the root formation takes place. You have to cut it here. What will happen? It will be transferred to the favorable condition in any other part of the plant. So next one is called as I told air layering. The parent plant is here. Here there is a stem continuity is here, but at the same time we remove the bark in this area. We remove the bark tissues removed. After that, moist soil or something else we have to place it in it here, and we tie with the polythene pack, and you have to put the holes because aeration is very, very essential for the root formations. But at the time, what will happen here? The hormones as well as other components also transfer this way, because the center part, what happened, xylem and phloem is there. Automatically, xylem conducting water and minerals, and phloem conducting the food materials. Okay. As a result, the root originated from the upper part of in this area. There is a root formation takes place in this area. The root also produce. Okay. After the formation of roots, we can remove the covers and made a cut here and placed in the favorable condition. This is also called as air layering. Okay. So go to the next one. Grafting. So grafting is a practice in plant. They do not root easily or have a weak root system. Suppose that any one of the plant it has a weak root system. That plant do not root easily or have a weak root system. You can practice this type of grafting methodology. It is successful in plant that have cambium for secondary growth. What is by cambium? What is by secondary growth? Already I told you, xylem and phloem is a secondary growth, otherwise called as cambium. The presence of cambium only, the water molecule as well as food material transfer from the one place to another place. Okay. Here you must note it down. 2 things is very, very important. Already they asked in the original question paper, NEET, particularly the basic or the main part is called a stalk. Which one is called a stalk? The parent, which one bearing the root? That means take an example of pot. Okay, the pot plant, the upper portion completely removed. Okay, remaining portion is called a stalk. Okay, so which one it has a root? Okay, that plant is called as what is the stalk. And which one we are going to graft the upper portion, any one of the plant, whether it may be good quality of the fruits or flowers or whatever it may be, that portion is called as what is cyan. It is also asked so many times. The stalk already asked in the 2018 question paper, NEET. Okay, see, one by one. So which one is called a stalk? The lower part of the plant. It's also connected with the root. Without root and the upper portion of the shoot system, it is also another plant, the sample A and the sample B, like that you consider. Okay, so you have to make a tie within this along with you have to tie with the two plants and make it as a one. As a result, you will get the good quality of the fruits or flowers or whatever it may be. It is used for the commercial purpose. Okay, it is also another thing. See, which is generally taken from the plant having a superior characters. Which one? So when you are going to select the plant, how will you make it? Plant having the superior characters. Already we discussed it. Take an example, say sapota or mango or grapes, whatever it may be. The fruit size will be very small one. Okay, some of the plants, suppose take an example, A plant, it has a small size of the fruits. At the same time, B sample, otherwise called as B plant, it contains the size will be big one, but the taste will be very less. A sweet taste will be very less, like that you consider. Okay, you have to cross these two things. Okay, which one? The small as well as the large one, and also these are the two superior quality. Which one is the superior quality here? The sweet taste is a superior quality of the A sample, and the size of the fruit is a quality of the superior quality of the B samples. You crossed it or grafted it, what will happen? You will get the new one. So both the quality will get it. So yeah, okay. So that is the another things. It is practiced in mango, apple, bio, citrus, guava, and rubber plant also. They are all considered it is commercial purpose. Okay, because we need fruits in good sweet taste also. At the same time, the size of the fruits also very big in nature. We are all searching that type of fruits only. Okay. In grafting, two plants are used to develop a new plant with combined triads from two parent plants. The cyan is the part of above ground level of one plant. The cyan is attached to the stalk, which is the rooted part of the second plant. Here you must note it down. Which one is stalk? Stalk is nothing but the rooted part of the second plant. Which one is cyan? Cyan is nothing but the above part of the stalk. Like that you have to imagine. Okay. As a result, you will get a single plant. Okay. The quality also you will get it. Okay. See here, you must see the diagram, then only you can easily understandable. See here, this is the first one. The stalk is also taken from the sample A, like that. Imagine with the root. This one is called a cyan. Cyan, it has two branches of leaves already. Any two nodes and also leaves also present in it. It is also a B sample plant. Both are crafted, okay, like this, and also wrapped with the waxed paper like this. After few days, what will happen? It will be grows entire plant and also gives you fruits as well as the flowers used for the purpose. Okay, this is called as what is that? Grafting. Here you must know which one is stalk, which one is cyan. It's very, very important things. Okay. What is the next one? The purpose of grafting. What is the main purpose of this grafting? Is to combine one plant quality of flowering or fruiting cyan plant with the another plant which has a quality or vigor and resistance. This is also another thing. One plant it has a resistant power and another one is not having the resistant power. Both are what is that crafted together. As a result, you will get the resistance of resistant plants. Likewise, okay. In most cases, the plant must be closely related, usually of the same genus. It is very, very important. Suppose they may ask the questions. It may be in the same genus or different genus. For example, prunus to prunus. Suppose when you are going to take any one of the plant, okay, Malvaceae or Solanaceae. Solanum tuberosum is nothing but a tuber. Solanum melangina, Solanum dorvum, Solanum xanthocarbum. These are all considered getting Solanaceae family. We have to take the same genus. Suppose when you are crossing the two different genus, what will happen? You will not get the positive result. It may be a negative one. Okay. See, likewise, in our potato is also one of the Solanaceae family, and tomato is also one of a, what is that? Solanaceae family. These two are crossed. Okay, you will get the new variety. It is a miracle. It is a miracle. They will get a single plant. The lower point you will get the what is the underground stem modification potato. The upper plant you will get the tomato, like that. Likewise, here also the plant must be closely related, usually the same genus. For example, prunus to a prunus or a malus to a malus. Okay, that is very, very important here. You have to know these are the 3 points is mostly asked in the NEET question paper. Okay, go to the next one. Okay, these are the things. Okay, so bud grafting. Here is a bud is also placed into the leaf. You have to make a small cut. The bud is also inserted. So as a result, the root formation takes place. And also another one is called as approach grafting. These are the two plants. One is called as a tree-like structure. Another one is called as a pot-like structure. So both are what is that grafted together. And then tongue grafting is also another one. You have to make a cut here, just like a tongue. And above stalk as well as the cyan is also there. You have to make a cut like as a what is that tongue, fix it together. Okay, fix it together and until and make it as a single plant. Then only it will be grow. Okay, and then growing craft, crown craft. Sorry, crown crafting. Crown crafting means this is considered as a stalk, and then this two is this two comes from which one? The another plant pot, another plant pot, otherwise called a sample B. It is called a cyan. See that the bark is slightly cut, made a cut like this, and inserted the cyan in this area and tie it. You will get the entire plant. Likewise, here also wedge grafting. These are the type of graftings made by the human being. That's why it is called as what is that? Artificial vegetative propagations. See. And then these are all very, very important things. At the same time, some of the root setting hormones, simply called as what is that? RSH, simply called as RSH, root setting hormones. Okay, first one, see the first point. Root formation can be promoted by the pre-treatment with the root setting hormones before they are inserted in the rooting medium. About some of the plant, they will not produce a root very easily. That time, what they will do, they are using some, what is that? Root setting hormones. Okay, 1 or 2 days in commercial purpose or laboratory work, they will do like this. Okay, they place it in a specific medium. That medium containing nutritive substances. The nutritive substances utilized by the root, and also it produces so many roots, and also followed by the shoot system also. This is also one. Likewise, the second one is also RSH can be made from a solution of active chemicals. Okay, this one is called as active chemicals. Already you know about what are the phytohormones. What are the phytohormones? Already you know about, mainly used for the growth of the plant, particularly tissue culture, and take an examples of and protoplasm fission, so on so. Okay, and even taken examples of the E. coli. Okay, commercial biotechnology also they were used. Which one, due to be substances that is called as agar. Okay, sir. Okay. Here, RS sketch can be made from the solution of the active chemical substances. That chemical substances here mentioned. What is that? They have given us two things. Is that one is called as indole butyric acid. It is an alternative form of indole acetic acid. The indole acetic acid, otherwise called as, what is that? What is that? Indole acetic acid. Anybody else? Indole acetic acid is also called as what? The first isolated phytohormones.
So is it? I'm not sure, but is it gibberellic acid?
No.
Cytokinin.
No. Indole acetic acid is also called as axin. Okay. Axin is the first isolated component. Okay. So alternative form, this is that is natural hormones. It is a, what is that? Artificial hormone. It is commercially prepared because they were used the butyric acid, indole butyric acid. Okay. So that's why it is called as, what is that? Chemical solution. Okay. Naphthalene acetic acid, NEAA. That one is called as indole acetic acid, IAA. But here IBA and also NEAA used for the root setting hormones. So these are the very, very important things. So they may ask so many times in this area also. So the root setting hormone, given examples of root setting hormones. Indole butyric acid, they may give indole acetic acid, gibberellic acid, and also indole butyric acid, and also indole butyric acid and naphthalenic acid. Like that they may give. So you have to carefully read the question and analyze the question and answer the questions. Okay, go to the next one. See, what are the significance of vegetative reproductions? What are the significance? What is the main role of this one? Vegetative reproduction, why we are doing these things, 2 points has given here, but there are so many points here. Okay. Plant in which usefully characters of a parent have to be maintained or propagated vegetatively. Normally, which plant they can easily propagated by vegetative, that type of plants we can easily, what is that, propagated by this method. And also another one is called as plants with reduced power of sexual reproduction. Some of the plants, they do not have the sexual reproductive ability. Okay, this is completely stunted in nature. Take an example of banana. When you are planting the rhizome, what will happen? It will be produce a pseudostem, and finally it produce the entire plant, isn't it? Okay, suppose we need more number of plants. There is a seed is there, but suppose when you are planting the seed, it is also stunted in nature. A staminode is staminode, like that, it is present in it. The anthers, the seed is placed in the favorable condition, it will not grow. It will take some more times. Okay, when you are planting the rhizome, you will get the yield within the 6 months. Sorry, 2 years. Okay, because banana is a bicarpic plants. Okay, and another one is those are loses their reduced power of the sexual reproductions. And second one is the long dormant period of the seeds. What is the meaning of long dormant period? Take an example of lotus. Okay, it has a long dormant period. Even take an example of so many pollen grains also having a dormant periods. For example, when you are going to take a crude oil or petrols, crude oil contains so many pollen grains is there. The pollen grains are inactive in nature, at the same time dormant in nature. Okay, they may be placed into a, which one, favorable condition, it will be grows a new plant. Okay, so dormant means resting stage. Once again repeat for you, normally all the seeds are having a, which one, reproductive ability, but some of the seeds having the, what is the dormant stage. Even beans or mungo, so whatever it may be, so many seeds are there. So many seeds are dormant period in nature. Such type of plants we can easily propagate with the help of vegetative reproduction. And also another one is called as poor viability. Poor viability means, it is also same. When you are placing the seeds, what happens? It cannot grow easily. Take an example, sapota. So yeah, okay. When you have planted this sapota plant, what happens? You will not get the yield within short period. Suppose you have to made a cut, that is called as cutting, stem cutting. You will get the yield within short period. Such type of reproduction done by the vegetatively. Okay, significance of vegetative reproduction given here, not all text, and also you have to add the which one, you know, the disease free, superior quality, all those things you have to add here. Okay, okay. Go to the next. Any question in this area, students? Any questions?
No, sir.
Okay. Next one is past. Until this, which one we have to discuss? We have discussed, you know, vegetative reproduction. Second one is called as asexual reproduction. It is also very, 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 very important things. Asexual. Asexual is nothing but the absence of sexes. Okay. Based on whether there is a participation of one organism or two in the process of reproduction, it is of two types. Asexual method is also one of the reproduction. That means absence of sexes. Here, maybe participate in, what is that? Only one parent only involved in this process. Such type of reproduction is called as asexual reproductions. They may be divided into two types. See here. When offspring is produced by a single parent, when offspring is produced by a single parent, whether it may be male, whether it may be female, with or without the involvement of gamete formation, that is very, very, very important. The production is called as asexual reproduction. Asexual means absence of sexes. The sexual reproduction means, what does it mean? Two parents involved in. That means opposite sex or opposite gametes. Okay. So when the two parents of opposite sex participate in the reproductive process and also involve the fusion of the male and the female gamete, it is called as sexual reproductions. These are the two things is that now we are going to discuss asexual reproductions deeply. Okay. Okay. Go to the next one. Asexual reproductions. See the asexual reproduction is common among the single cell organs. Will you please tell me any single cell organism here? Already mentioned here. Paramecium, Euglena, particularly tell us. Yeah, very good. Super. Unicellular eukaryotes. They are all protista. What is the meaning of protista? What is the meaning of protista? What is the meaning of monera? Simply we may call as what is meant by eukaryote, what is meant by prokaryotes. Yesterday also we discussed. Anyone else? What is meant by eukaryotes? What is meant by prokaryotes? Pro means primitive, karyon means nucleus. It is not well developed nucleus present in it. At the same time, cell bound organs completely absent. But in this category, eukaryote means, what is the meaning of eukaryotes? The well developed nucleus is also there, rigid cell wall is also there. At the same time, they are having all cell bound organs present in it. Endoplasmic reticulum, smooth and rough, mitochondria, Golgi apparatus, and ribosomes, dictyosomes, and also vacuoles, all those things all present in the. So protista, they may ask the questions. The protista, paramecium also comes under the getting known as protista. Paramecium, protista, Euglena, protista. What is the meaning of that one? You know, it is also comes under the getting unicellular eukaryote. Okay, we are all are considered the getting known as multicellular eukaryote. We are having more than millions of cells in human being and animals. Okay, particularly single cell, that is protista, particularly eukaryotes. Okay, and monerans. What is the meaning of monerans? Monerans is nothing but the unicellular. Prokaryote, already I told you what is meant by prokaryote. Example, bacteria. Bacteria is also one of them minute microscopic unicellular prokaryotes. You can see only under the microscope only. Okay, the organism or the parent cell divide into two to gives rise to a new individual. So you know about the bacteria, how it be reproduced. There are so many methods there. One is called as what is that? Binary fission. Second one is called as conjugation, and also another method also there. Okay, so these are the things. Thus, in this organism, cell division itself a mode of reproduction. Take an examples. During the cell division, what are the physiological changes which occurs in plant as well as animals? You know, Rilbert, there are two things is there. What are they? Will you please? Anybody? During cell division, what will happen in the plant or animal cells? Anybody else? What will happen inside?
So,
Yeah.
so so the cell starts splitting, sir, from the nucleus.
Okay. First, whether it may be a cytoplasm or whether it may be a nucleus, first division takes place. Which one is the first divide first?
So the first division will be the cytoplasm, sir. So
Okay.
so the nucleus
No.
divides and then it
Yeah.
goes into the other side, and
Yeah.
the cytoplasm splits, and then
Yes.
the formation of in if for
Good.
example in plants it's cell plate formation, and in the animal cells there's the splitting of the cells, sir.
Okay. Good. Super. But first one is called as nucleus divided in two halves. Then what happens? Cytoplasm automatically divided. Okay. Then only the two daughter cell you will get it. Okay. Okay. Before that, just see the what is that chart? Already we discussed. See, asexual reproduction is also what are the first one is called as binary fission and multiple fission, budding, fragmentation, sporulation, vegetative propagation. Vegetative propagation already we discussed. Natural and artificial. Natural contain root. What is that? Root also undergoes a vegetative propagation, stem and leaf also there. And artificial already we discussed cutting, grafting, layering, and also tissue culture. Tissue culture we didn't discuss, but slightly we saw it, isn't it? Okay, we will discuss at the time of biotechnology and biotechnology. Okay, see the next one. Asexual reproduction, already told you. In this method, a single individual is capable of producing offspring. Only one parent is involved. That is called as, what is that? Single parent. As a result, the offspring that are produced are not only the identical to one another, but are also exact copy of their parent. So this is very, very important. Most of the cases, what happened? No, it is not identical to the one another, but are also exact copy of their parent. This is the question. You note it down? Okay. Are these offspring likely to be genetically identical or different? Genetically identical or different? You must know the answer. The term clone is used to describe such a morphological and genetically similar individual. What is who did this? See, the iron Wilmot, you know what about the iron Wilmot? One of the scientists, 1996, did the work. Okay, the Dally goat. Okay, the Dally goat is also one of the asexual reproduction. He took a single cell from the only one parent, and also undergoes a process. You know about the reproduction process. As a result, he got what is a two Dally goat. After 6 months, what happened in lead to death? Why? No, because the immunity power is lost there. That's why. Okay, sir. Okay. The type of reproductions already you know asexual and sexuals. Asexual, what are the things you know? Already we discussed. See here, fission, budding, and also spore formation and regeneration, vegetative propagation. We are going to discuss very deeply. Okay. See the next one. This is just intro only. This short only for the intro. So go to the next one. See, there are few common method of asexual reproduction. What are the few common method? You know, first one is called as fission. Second one is called as spore formation. Third one is called as budding, and fourth one is called as fragmentation. These are the four has given in your book. You have to discuss one by one deeply. What is by fission? What are the example for the fission? See here how they are fissions are classified. Okay, See the fissions. In fission, and first nucleus divided, I already told you. Okay, first and cytoplasm next. During the time of reproductions, okay, take an examples of amoeba, paramecium, euglena, or whatever it may be. Okay, the first division is, what is that? Nucleus undergoes the divisions. Next is the cytoplasm, is the next one. That is called as, what is that? Karyokinesis first and cytokinesis is the second one. Okay, so as a result, what happened? Mother cell split into two equal sized daughter cell. This is referred to as the binary fission. Bi means two. A single mother cell split into gives two daughter cell is called as binary fissions. Here, what will be the main physiological process? First division is called as karyokinesis and second division is called as cytoplasm. Okay, the fissions also classified into, okay, according to their divisions, whether it may be transfer or longitudinal or more than one, or and unfavorable condition, they produce some specialized, what is that, spore. Okay, likewise, they may be classified into 5. Okay, one is called as simple binary fissions, and the second one is called as transfer binary fissions. Third one is called as longitudinally binary fission, and the fourth one multiple fission. The last one is encistations. Simple binary fission, given examples, no, amoeba. And transfer means it is also fission takes place in transfer axis manner. Then only it will be split into two. Example, paramecium and planaria. But planaria, it has a specialized feature. Will you please? Anybody else? Planaria. Any one of you? Will you please? Planaria. It has a specialized feature. Will you please? Nobody else? Okay. Okay, I will tell you. Okay, planaria has a highly regeneration capacity. Highly regeneration means, what is the meaning? Suppose any part of the animals, what is that? Cut. Okay, it will grow as a new one, similar to that of the parent. Okay, not only this is planaria and also starfish and so many things is there. Okay. And longitudinal binary fissions, there is examples euglena and vorticella. It is also longitudinal axis. It gives it. This is the parent and divided into longitudinal axis. As a result, you will get the two binary, what is that, daughter cells. And multiple fissions, here the specialized single cell, otherwise called a single, what is that, mother cell, divided into more than one within the, what is that, mother cells. But here, what happened at the specialization is here, there is no, what is that, cytoplasmic divisions. Only the nucleus divide into more than 10 or 15 like that. Okay. Here, only the karyokinesis only takes place, but not cytoplasm. After it comes out, the cytoplasm gather it and also form as a new one. That is called as multiple fissions. Amoeba, plasmodium, and monocystis. These are the examples. Okay. And encystation is examples. Amoeba. What is the meaning of encystation? Means cystation. Cyst. You must note it down. During unfavorable condition, take an examples of high temperature or whether it may be a cold temperature. Any one of the, just like that, amoeba, like the microorganisms, what happened, it produces the, what is the outermost covering. It's a hard coat like that, that is called as cyst. Okay. Okay. So these are the, what is by fission here. So this is nothing but one of the, what is that? If split into gives two equal size according to the nature. One is called a simple binary fission. This one is also called a simple and transfer, longitudinal and multiple fission and encistations. So these are the things. Okay. See, sometimes the nucleus divides several times by the amitosis. Okay, what is the meaning of amitosis? Maximum sexual reproduction and asexual reproduction and vegetative reproduction also takes place. And sexual is only in which type of cell division? Meiotic. Okay, but asexual as well as the vegetative reproduction mostly takes place in the which type of cell division? Mitosis. Okay, here several time by amitosis to produce. Amitosis is nothing but only the nucleus will be divided, but cytoplasm is not divided here. To produce the many nucleus without involving any cytokinesis. Without the cytokinesis, many nucleus formed within the parent cell. Such type of division is also called as what is that? Last one we discuss, multinucleated, otherwise called as multiple fission. Okay. Later, each nucleus gather a small amount of cytoplasm around it, and the mother cell split into many teeny daughter cells. Example: amoeba, plasmodium, and monocystis. In course of time, each of these daughter cells starts a free life and transform into adult individuals. This kind of fission is called as multiple fissions. Okay. So today, what are the things we discuss? What is meant by asexual reproduction? What are the type of asexual reproduction is there? Okay. See, one by one already discussed. See, these are the things we are discussed. What is meant by asexual reproduction? What are the type of asexual reproduction is there? What are the significance is there? Okay, cutting and layering, grafting is also. What are the type of cutting? What are the type of grafting? And also another things is also there. And root setting hormones. What are the main significance of vegetative propagation at the same time vegetative reproductions? Okay, asexual reproduction. What is the meaning of asexual reproduction? What is the meaning of sexual reproductions? Okay, and then so asexual and sexual. What is my protista? What is my monera? Otherwise called as what is that? Eukaryote. What is my prokaryote? Mitosis and meiosis. Okay, and what are the type of propagation that is particularly vegetative and natural and artificial? These are all comes under the getting whether it's natural or artificial. Okay, what are the type of? It is a asexual reproduction, fission, and spore formation, budding, and fragmentation. And we discussed fission only. Okay. Any questions, students? Do you have any questions?
No, sir.
So this is binary fissions examples. Say the bacteria is this is the parent cells divided. First, which one is divided here? The first see it here, nucleus is divided, and then after that there is a cytoplasm division takes place. As a result, you will get the two offspring of the daughter cells. Okay, this is called a simple binary fissions. Okay, the parent divided to two gives two daughter cells, exactly similar to that of the parent. Okay, for the next one, see that this is called as amoeba. Okay, amoeba means everybody, it's also one of the microscopic unicellular. What is that? Prokaryote. Okay, sorry. So listen here. Here there is a nucleus is there. First division takes place in there. Here also division divided into two cells, and also the cross. What happened? The cross wall formation also takes place and split into two daughter cells. This is the new daughter one. This is also binary. Binary means two divisions. Okay, already know about. This is called as bacteria. Bacteria is also undergoes the undergoes the divisions. See here, nucleus division first takes place, and the cross wall groove-like structures also originated from the root. Cell wall. So as a result, what happened? It may be divided into two and separated. It's also undergoes the further division. It's similar to that of the cyclic manner. Okay, when it undergoes the maturation process. Okay, see the next one, binary fission of the paramecium. Paramecium is also another one. Already in our body is similar to that of the sleeper-like structures. Okay, and also it is also having the nucleus and other cytoplasmic cellular organelles, bound organelles also present in it. The division is also takes place in our body, divided into two and split into the two daughter cells. And when it enter in the maturations, it enter into the first one. Okay, and then so the protista. What are the examples? These are the examples. Protista is nothing but the unicellular eukaryotic. Most of the cases they ask the questions. Most of the question paper you can see these type of things. Protista. Example, eukaryote. These are the protista. What are they? Slime mold, amoeba, euglena, dinoflagellate. Dinoflagellate means flagella. Flagella is one of the most important locomotory organs. Here, how many flagella present in it? 2 flagella is there. Okay, that's why it is called as dinoflagellate. Paramecium already we discussed. Diatom is otherwise called as golden algae. Okay, and macroalga means which one is that? It is which is biggest one. Okay, macro means it may large size in nature compared to the other algae. Okay, and then see type of binary fissions. Amoeba, irregular binary fissions. So comes under there are 2 different kinds of binary fission is there. One is called as irregular binary fission, and second one is called as longitudinal. Longitudinal axils divide into 2. First nucleus division takes place, and also the cytoplasm again. Next, here the transverse means it's a vertical. This is longitudinal. This one is called as vertical, and also oblique binary fission. In any part, any sides, it can be split into difference. These are all called as the type of binary fissions. Okay. So same things is here. This is called as binary fission. These are all called multiple fissions. Multiple fissions means what is the meaning? There are so many things as given here. Plactomycetes. It's okay. And also another one is called as sterine area. Okay, and Eucyposicum, like that they have given here. Okay, just you know this is the daughter. I'm sorry, this is the mother cells. The mother cell, there is a nucleus. The nucleus division takes place without which one? Amitosis. Amitosis takes place inside. As a result, what happens? There are so many nucleus divided into number of copies. Okay, and finally, what happens? It comes out. These are the matured daughter cells. Then it also undergo the further process. Okay. So reproduction, binary, conjugation, autogamy, cytogamy, and endomixis. I will tell you one by one these things. We will see in the next class. Any question, my dear students, up to this?
No, sir.
No. Are you clear about the concept?
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
Thank you so much for your kind support and for the participation. Thank you. Sir, Mahendra sir.
Yeah, you can make me host. Yeah, thank you. Yeah.
Thank you, sir. Thank you. Shall I finish it, sir?
Yes.
Okay. Thank you.
Thank you.